Hello. Today I will be talking about the genetic disorder known as achondroplasia, specifically its genetic basis, symptoms, and treatment options. After completing this lesson, students should be able to explain the genetic mutation responsible for achondroplasia, identify key symptoms and affected tissues in individuals, and evaluate current treatment options. At the bottom of the screen, you will see an image of a child with achondroplasia on the bottom right versus a child with average stature on the left. There is also a picture of the FGFR protein structure, which we will discuss later. Skeletal dysplasia is a group of rare genetic disorders that affect bone and joint development and can also impact cartilage and neurological function. As seen in the image here, achondroplasia is only one of these disorders, occurring in around 1 in 25,000 live births, so it's still a rare disorder. Achondroplasia affects both males and females equally and occurs in all ethnicities and racial groups. The inheritance pattern for achondroplasia is autosomal dominant, meaning the child only needs to receive one copy of the mutated gene to develop achondroplasia, as seen in the image here. However, this only makes 20% of the population affected with achondroplasia. The rest arise from de novo or new mutations not inherited from parents, with a significant risk factor being advanced paternal age. The gene responsible for achondroplasia encodes for one of the key receptors in regulating chondrocytes or cartilage-forming cells. This gene is fibroblast growth factor receptor 3, or FGFR3. The protein is a transmembrane protein waiting to be activated by a specific ligand binding. This specific ligand is fibroblast growth factor, or FGF. Once bound, this starts a signaling cascade that activates MAPK and STAT pathways, as shown in this image. These pathways negatively regulate chondrocyte proliferation and differentiation, essentially telling the cells to stop growing. The most common mutation leading to achondroplasia is G380R, a missense mutation where glycine is replaced by arginine at position 380 of the FGFR3 protein. This mutation leads to constitutive activation of FGFR3, meaning the FGFR3 receptor becomes activated without the need of FGF binding, thus resulting in excessive inhibition of chondrocyte proliferation and differentiation. This leads to distinct phenotypes. While the disorder affects nearly all the bones in the body, it is most obvious in the limbs, and on average, people with achondroplasia will end up 25% shorter than people of average height. Here I have shown an image showing some distinct phenotypes associated with achondroplasia, which include short stature, disproportionately short arms and legs, large head size, frontal bossing, spinal curvatures, trident hand, bowing of legs, short metacarpals, and flattened nasal bridge, to name a few. To understand why those symptoms are seen, we have to take a closer look at how bones grow. In people, there are two different ways that bones form, but the type of growth that is affected in achondroplasia is called endochondral ossification. This is the main method responsible for lengthening bones. This process involves several stages, the first of which is the formation of the cartilage model. In this stage, mesenchymal cells differentiate into chondrocytes, forming a hyaline cartilage model of the future bone. These chondrocytes proliferate and form columns, which expands the cartilage model. Chondrocytes grow larger and secrete the extracellular matrix, preparing for calcification. The primary ossification is formed when the cartilage matrix calcifies and blood vessels start to invade the cartilage, thus bringing osteoblasts, or bone-forming cells, that replace the cartilage with bone, usually starting in the center of the bone. Secondary ossification centers form at the ends of long bones. The regions between the primary and secondary ossification centers, known as growth plates or epiphyseal plates, continue to produce cartilage that is subsequently replaced by bone, contributing to the lengthening of the bone. In achondroplasia, the mutated FGFR3 gene leads to overactive receptor signaling, as was discussed earlier. This severely limits the proliferation and hypertrophy of chondrocytes in the growth plates. This results in decreases in the division of chondrocytes, as seen in the top layers of this image. This leads to shorter columns of cells. Chondrocytes are not able to grow due to shorter columns, thereby limiting the expansion of the cartilage model, as seen here in the second layer. Thus, the cartilage model is not adequately replaced with bone. This is an oversimplification, but the result is premature closure of growth plates and disproportionate bone growth, as seen in the phenotypes. Although there are various treatment options, some of the key ones I wanted to highlight are growth hormone therapy, which involves the administration of recombinant human growth hormone to promote growth in children with achondroplasia. It may increase height velocity, but the long-term impact on final adult is variable. Vosoritide, a CMP analog, improves bone growth by targeting the FGFR3 pathway. It has been recently approved for children, showing promising results in increasing growth velocity, but requires daily subcutaneous injections. Limb lengthening surgery is a surgical procedure involving the lengthening of the bones in the arms and legs. It helps increase stature and improve the proportionality of limbs. It can significantly increase height, but requires multiple surgeries and has a long recovery period. I have included an image here of an individual affected by achondroplasia who underwent 4 years of limb lengthening surgery to grow from 3 feet 9 inches to 4 feet 11 inches. Lastly, I wanted to highlight spinal surgeries. These are procedures to correct spinal stenosis and other spinal deformities such as kyphosis. These help alleviate pain, improve mobility, and prevent neurological complications, and are often necessary due to common spinal issues in individuals with achondroplasia. To conclude, achondroplasia is a genetic disorder with significant impact on bone development. Today, we reviewed the genetic mutation and mechanism responsible for achondroplasia, identified key symptoms, and analyzed how normal tissue versus altered tissue happens in individuals with achondroplasia. And lastly, we went over treatment options. Thank you. Here are my references, and have a nice day.